Now we turn to the diversity of the bacteria and archaea. Our discussion here will be woefully brief. Many of you will discuss the subject in some detail in microbiology courses. How many species of these organisms are there? This is a very difficult question to answer for several reasons. For one, the most convenient species definition, the biological species concept, doesn't apply to organisms that don't reproduce sexually. The morphological species concept, or defining species based on similar appearances, not much help either, given the few types of cells that we can see through the microscope. Biochemical tests have been helpful since the dawn of scientific microbiology. For a long time, we could only research and describe species that we could culture, but the revolution in sequencing nucleic acids, in addition to demonstrating a colossal rift between bacteria and archaea, have revealed the enormity of microbial diversity that we are unable to culture on petri dishes. As the difficulty and cost of DNA sequencing have plummeted over the last few decades, as well as the increase in computing power to analyze these data, we have discovered vast reserves of biodiversity, previously undiscovered phyla of bacteria and archaea, in just about every habitat where the lens of science is focused. This process of taking environmental samples from unique habitats and examining the biodiversity solely from DNA sequences has enabled researchers, including some here at Auburn University, to make discoveries of useful products such as new antibiotic drugs. This process is called metagenomic prospecting. If we look at a phylogeny of the prokaryotic lineages, we revisit one of the first problems that I talked about. There is no way to make a monophyletic grouping of the archaea and bacteria. The last universal common ancestor of cellular life, also known as LUCA, is represented at the root of this tree. But we cannot cut out the prokaryotes without making a second cut to prune the eukaryotes out. At best, prokaryotes are paraphyletic. They may even be polyphyletic. The relationships of these organisms date back so far, a few billion years, that LUCA's identity is the coldest of cold cases. Starting with the domain archaea, some microbial taxonomists say there is only one kingdom. Others define 4 or more kingdoms. This distinction is not very important for us. You should know that the archaea are entirely gram-negative. They do typically have cell walls, but instead of peptidoglycan, they possess pseudopeptidoglycan, a different biological polymer of proteins and carbohydrates. The first recognized archaeans were all extremophiles, or found in growing in habitats at the most extreme of conditions, hottest, saltiest, most acidic. Least oxygen tolerant? Perhaps this is something we see in the archaeans known as methanogens, which produce methane gas, CH4, as a metabolic waste product. Methane gas, by the way, is odorless. If you smell something like rotten eggs or flatulence when you find yourself in a swamp, that is a different respiratory waste, hydrogen sulfide. As we've delved into environmental samples of unculturable microbes, it turns out that archaea can grow in non-extreme environments also. One place no archaeans have been found is acting as the primary agents of disease, though they can be important symbionts, including mutualists. No archaean pathogens. The principal lineages of archaea are shown in this phylogeny, though we may discuss another important group as a class. These 4 kingdoms, if we allow, are the Euryarchaeota, the Crenarchaeota, the Corarchaeota, and the Nanoarchaeota. While I've included a few facts on this slide, just being acquainted with these 4 names will be adequate for our purposes at this time. Moving on to the diversity of the true bacteria, or the eubacteria, I'll show you this very stripped down phylogeny from our course textbook. As with the phylogram of the archaea, this is a woeful underrepresentation of the diversity of the bacteria. When and if you take microbiology, you will see. Again, for our purposes, the number of kingdoms within the domain is subject to the opinions of different microbiologists. As many as 17 different kingdoms have been proposed, but let's just go with one. And we'll call that one kingdom of the domain bacteria, eubacteria, or the true bacteria. Looking at this simple phylogeny once again reveals 5 clades of bacteria. Proteobacteria are further delineated into 5 clades, each given a Greek letter prefix. I mentioned alpha proteobacteria when I spoke about endosymbiosis. Mitochondria are derived from within alpha proteobacteria. Chlamydias and spirochetes share a clade. Chlamydias are best known for causing a sexually transmitted infection, though there are many other gram-negative species in diverse habitats. Spirochetes are known for 2 things, mainly being spirilliform, or spiral-shaped, and for being important pathogens, such as those that cause syphilis and Lyme disease. Cyanobacteria were once known as blue-green algae, although that's not a name we hear very much anymore. It's because of their color and because they're photosynthetic. It's thought that cyanobacteria were some of the first organisms to perform oxygenic photosynthesis. That is the photosynthesis that produces oxygen gas, and the same photosynthesis we see in plants and eukaryotic algae. There are many colonial cyanobacteria that form macroscopic mats. Gram-positive bacteria include both Streptococcus and Staphylococcus, important human pathogens, and are so called because of their reaction to the dye crystal violet in the gram staining procedure. Sharing a clade with the gram-positive bacteria are the mycoplasmas. Mycoplasmas have no cell wall, which is why they look more undefined in their outline. They're not really cocci, spirilla, or bacilli. Mycoplasmas are resistant to many antibiotics whose mode of action is blocking cell wall formation because they don't have any cell walls. Our tour of prokaryotic diversity is very brief. Many of you will gain further insight by taking a course in microbiology, and there are a lot of known unknowns in the microbial world. As you can see in this phylogram of cellular life from the Interactive Tree of Life project, microbial diversity dwarfs eukaryotic diversity. However, our main focus this semester will be the diversity of eukaryotic life. We just need to learn a few things about the archaea and bacteria before we go there. So to close, once again, here are our learning objectives. And next up, the eukaryotes.